Hello.
Oh.
Well, we're really glad all of you are here. So thank you for coming to what's now becoming, I guess, a pretty regular thing for us to have Wednesday events. And so we're very glad that you're here because this is a rescheduled event. So we're doubly glad that you said you would come the first time and that you're actually here once we've gotten it back on our calendar. So my name is Ann Merchant and I work for the National Academy of Sciences.
I'm Rick Lovard. I'm the Program Director of the Science and Entertainment Exchange, which is a program of the National Academy of Sciences.
Yes, we actually work for the same place, which is why we're doing this together. And so my job at the top of this is always to say a little bit about the National Academy of Sciences. If you've been coming regularly, you probably know this by now, although I think we're often confused by many people as being part of the federal government. So the first thing I always say is that we are a private nonprofit institution in Washington, D.C., created in 1863 so that the federal government would have a source of unbiased, evidence-based science to which they could turn when creating good policy. And so we continue to do that today. There are a couple of studies that I wanted to mention that I think are very relevant to what's happening today. One of which I mentioned briefly last week, which was quite apropos for the topic, but I think it was only on Tuesday that the Academy formally announced that they had convened their committee on vaccine allocation. This was requested by both the CDC and the National Institutes of Health, and so they have pulled together a very august group of experts to think about the ways in which vaccines should be allocated once they do become available, taking into account a variety of factors such as geography, the most vulnerable populations, individuals at the highest risk. And I think in particular, they want to make sure that communities of color are taken into careful account when the allocations are considered. So that probably will be coming out in the fall, early September, I think, something like that. So they'll be working hard on that. The other thing that we've just released is the way in which the COVID pandemic has been impacting cancer care. There are some things, obviously, now I, for example, right before we all were sent home, I had a slow-growing squamous cell carcinoma that I needed to have removed. Well, that's a very slow-growing cancer. It doesn't really have to be treated right away. But obviously, there are many cancers where that's not true. So how does something like COVID impact cancer care? So the Academy has released a report on that. And so if that's relevant to you, all of our reports are available for free download from the Academy's website. And I wouldn't say they read like a bedside novel, but on the other hand, they do all have executive summaries at the beginning, so that the conclusions and recommendations are all in the first perhaps 20 pages. So they're pretty easy to get through at least that part if you're not interested in reading the successive 400 pages. But as Rick said, the Academy, we have a responsibility for a wide swath of the sciences and providing input in many different topics, not just COVID related, although that tends to be a big proportion of our work right now. But the Science and Entertainment Exchange was created in order to give voice to a lot of that kind of good work that wouldn't ordinarily meet the needs of a variety of different audiences. And so Rick is the director of the exchange, and he'll tell you a little bit about what that is and what we do.
Hi, so the Science and Entertainment Exchange is a program of the National Academy of Sciences. If you are a writer, director, producer, creator of any sort of mainstream media content, and you have a question about science as you're creating that content, we will connect you with an expert in STEM for free who can hopefully answer your questions and can certainly contextualize them better than Wikipedia. Not to knock on Wikipedia. If you are a media maker and you do want to contact us, you can reach us over email or through our 844 number, not 800, 844-NEED-SCI. We've done over 300 events like this one. Well, most of them have been live and in person, but lately digital. And over 3,200 consults since we opened our doors about 11 years ago, including Avengers Infinity War, Man of Steel, Black Panther, Captain Marvel, and you can catch a recent release on Hulu in the form of Palm Springs, which is a time travel movie. And if you are a STEM professional and this is the first event that you've tuned into and you're just getting wind of what we do and who we are, please do reach out to us if you're interested in getting involved. We're always looking for people who are interested in being connected to storytellers. So quickly, a little bit about today's event. We only have the one speaker, but he is amazing. I can't wait to get him on, so we'll get through this as quickly as we can. But if you have a question as he's speaking, they're down here, it says Q&A. Some of you have already discovered this. Sorry we didn't have hold music to open up. I know it was a little disorienting to not have sound, and a bunch of you asked questions about that. So I will be watching as Neil is speaking today to try to field out as many questions as we can. And we have an excruciatingly short Q&A period for this event. If we don't get to your question, my apologies. We're gonna get to as many as we possibly can. And then some of you who did donate, and thank you for donating, also requested or were able to request a special VIP on-camera Q&A session that we're going to be doing afterwards. If you would like to do that next time, then just click the donate button when you RSVP. Just donate a couple of bucks, and we will make sure that you get into that VIP section the next time. So I want to thank really quickly Ameche, our AV person who makes this event happen and runs the whole thing really. Courtney Sloan, Jeff Fishman from the DC Creative Engagement team, who really help us behind the scenes more than I can say in 3 seconds. And Sachi Gerben, my colleague here in DC, without whom, honestly, the exchange would totally fall apart, and everybody knows it. Also, thank you to Gia Mora, who is live tweeting for us today. We really appreciate it. I don't have anything right now to tell you that she's doing right now in this time. I think her play was called Einstein's Girl. Forgive me if I'm wrong about that. It was. Okay, good. Gia, please tweet whatever you want people to check out. And finally, I got to thank our sponsors, Howard Hughes Medical Institute, without whom this event would not be happening today. Also, the Alfred P. Sloan Foundation, Walt Disney Company, Film Nation, Google, Lyda Hill Foundation, Corteva, and so many individual donors like you. We can't even believe how much support we've gotten from individuals for these events. And thank you. Every dollar that you donate does end up going back into more of these events. So thank you.
Yes, that's absolutely true. So, and we've made it a bit of a tradition to have a couple of little diversions, I guess we should call them, at the beginning of our program to talk about some things that we've been seeing online. One of which, and Rick thinks this is completely creepy, but I think it's really interesting. It's called window swap. And people have taken short videos of what they see out their windows. And there's a website on which this is aggregated. So you can go and you can click and you'll see a window in Helsinki. And then you'll click and you'll be in Hong Kong, and then you'll click and you'll be in Montana, and then you'll click, and it's just really cool. Some of them are in rural areas, some of them are in urban areas, some of them you're not really sure what you're looking at, but it's cool. I think I spent time, a friend of, actually it was Sunny Bates, last week's moderator, that told me about it, and then it was featured in the New York Times. So I highly recommend a window swap. I don't think it's creepy. I think it's fun.
Super creepy. It's super creepy.
Not creepy. There's a firefighter in Washington, D.C. His name is, I'm looking at Jim Mazzara. He is a firefighter for Engine Company 1 in Washington, D.C., and he's a bagpiper, and he's been going around D.C. to the hospitals, to different neighborhoods, to churches, and he's been bagpiping just to make people feel better. So, I mean, if I looked out my window and there was a bagpiper out there, it would make me feel better. I thought that was very cool. I also watched a short film called Father and Daughter. It's on YouTube. It was very sweet. It's about separation and loss, but it was very heartwarming and very touching. And if it's a little too touching in this time and you need something a little different, well, I also sat and watched about 45 minutes of outtakes and bloopers from Friends. So there's that. And Rick, because he brought a stuffed animal last time, I thought that I should bring my stuffed animal. So I have these little hedgehogs that I thought I should bring. So there, Rick, I have hedgehogs.
Anne made me a hedgehog too. And she may make one for you if you request it. I'm just saying.
Well, That's enough of that. So Neil Shubin has probably run away by now because he thinks, oh my God, they are truly insane. The National Academy of Sciences will never recover from this. And I should also mention that the National Academy of Sciences is an honorific institution. So the way that the academies was originally conceived is that there would be membership conferred upon the most illustrious and the most accomplished of our scientists. That remains true today. It is a very small group indeed. I think it's just over 2,000 people are members of the National Academy of Sciences. It's considered to be the highest honor in American science. And so we are very pleased that we have a member of the National Academy of Sciences on our virtual stage. And we've actually nabbed him before. We had him in the very early days of the program. And to my great chagrin, we scheduled him. And then I had to call he and Sean Carroll, both members of the NAS, and no staff member wants to do this. So anyway, we have to change the date at the last minute. It's not a big deal, right? And Neil was so gracious and said, no, it's not a big deal. I'll be there. So we are equally happy that he was able to make room in his schedule for us today. And so I'm going to turn it over to you, Neil, and let you have the rest of this time.
Well, thank you, Anne, and thank you, Rick, and thanks all of you for sharing your afternoon with me. I'm delighted to share mine with you. I'm a paleontologist, evolutionary biologist. I've been doing paleontology and leading expeditions for over 40 years. It scared me when I thought about that originally, but over 40 years. I've led expeditions near the North Pole. I've led expeditions near the South Pole, and pretty much in all 7 continents. But my lab also works on DNA, and we also work on the evolution of genes and genomes and so forth. And at some point, I was thinking about my career, and I was thinking about evolution and how we use fossils and DNA and understanding of embryos to really piece together the great transitions in the history of life, these great mysteries in the history of our planet and history of our species, in the history of all living things. And so that all came together in a book I recently wrote called Some Assembly Required, and this talk is sort of derived from that book. And the central message is, when you think about the great transitions in evolution, how birds evolved to fly, how fish evolved to walk on land, how humans with consciousness came about. These transitions seem so utterly impossible when you first begin to think of the endpoints. But when you collect fossils, when you look at genes, you find unbelievable surprises in the record of the history of life. So I'm going to start sharing my screen. I'm going to show some slides, and we're going to cover 4 billion years of evolution, sort of, fossils and genes, in 20 minutes. So let's get going. So this is actually one of my field sites in Antarctica from about 2 years ago. So that's the extremes we go to crack rocks to look for ancient fish. So as I was finishing the book, Some Assembly Required, and as I was thinking about evolution, I was doing some reading on the side, and I was reading an autobiography of this remarkable human being, Lillian Hellman. As many of you know, Lillian Hellman was a playwright, illustrious playwright, famously hard living. She was blacklisted by the House Un-American Activities Committee, the McCarthy era and so forth. And in looking back at her life, and just looking at that picture, you can imagine what that life was like. And looking back at her life, she had a quote that captured so much of her life, but so much of the way we think of evolution nowadays. And the quote that captured my imagination was, "Nothing, of course, ever begins when you think it does." That's the story of evolution in a nutshell. If you think lungs evolved as animals, evolved to walk on land, or feathers came about as animals evolved to fly, and other inventions came about during great revolutions, you'd be in really, really good company. You'd also be 100% wrong. And we've known that for over a century. And in fact, the more we study fossils, the more we study DNA, the more we find evolution could not work in any other way. So let's step back and let's think of where we are today and some of the great surprises that we've had from evolution. And again, returning to Lillian Hellman's quote as much as we can, let's think about what we've learned in the last several decades. And what we've learned so much in the last several decades is that, yes, we've had some amazing fossils, but we've also uncovered so much knowledge about DNA and proteins and how these things work inside cells and how they work to build bodies. Let me just open with a few surprises. If you think about DNA, you have a 6-foot-long strand of DNA crumbled and balled up in on itself inside the nucleus of every single cell of your body, in my body as well. And let's say we have 4 trillion cells, and each one of those contains a 6-foot-long strand that's been rolled up on itself. If you unrolled all the DNA in your body and put it end to end, it would extend from here almost to Pluto. That is an enormous amount of genetic information. It's remarkable. And we've learned so much about this in the past several decades. We began with the Human Genome Project a few decades ago that cost, several $billion, took a decade and a half. Now we have genomes for many thousands of species, and we can sequence a genome in an afternoon for less than $1,000. And there's so many surprises embedded in that. So many surprises about not only genetics, but also about our own evolution. And I just want to sort of relay a few of them, and then we'll return to fossils later on. And we're going to begin with one of the bigger surprises, actually, the one that sort of blew my mind when I first learned about it. And it begins with the work of this gentleman, Jason Shepard. So Jason is a neurobiologist at the University of Utah. And Jason's had a passion for his whole scientific career to understand memory. What's the genetic basis of memory? What's the physiological basis of memory? And there's so much embedded in that. There's so much of human health, human well-being, basic biology. It's a great question. So Jason sort of centered, like a good biologist, on a particular gene that he was really interested in. It was a gene called ARC, A-R-C. And the reason why is ARC is involved with memories. Mice that have a mutation in the gene ARC can solve a maze, and find cheese in the middle of a maze, but they can't remember their solution the next day. Humans who have a mutation in the gene ARC have all kinds of cognitive deficits. So this is clearly a gene that's heavily involved in cognition as well as memory. So what he did is he isolated the protein that the gene makes. Right? Sounds like what we usually do in science, and he popped it under a microscope. Now, that was a bit of a saga for him. I'm not going to tell you the whole story, but he popped it under a slide, and this is it. This is ARC gene. And the gene makes a protein, and here's an electron micrograph of that protein. So, you're looking at something that's about a micron long. And you see those little blobs in there? Like, bam, right there. See that little arrow pointing to a circle? He noticed there are these little blobs all over the slide. And these are clumps of proteins made by ARC. He looked at this and said, I've seen this before. And he dug out, I dug out a microbiology textbook, found this photo, and I have an arrow pointing to another clump that looks identical to the ARC one. Only this isn't a memory gene. What is this? This is HIV, the virus that causes AIDS. Jason was like blown away. They were identical. So he's working at a medical school at the University of Utah. He calls some folks in the building next door who actually work on HIV. And he took them to show them the slide that on the left. And he didn't tell them what it was. He says, hey, what do you guys think this is? And they're like, oh, of course, it's a HIV, virus that causes AIDS. And Jason's like, nope, sorry, that's HIV. I'm sorry, that's a memory gene, ARC. They're like, no way. So they went, what? And they went back and forth. And finally, they sequenced the gene. They looked at the detail of the protein. And it turns out that the sequence and structure of the proteins made by ARC, and indeed the gene itself, contains the classic signature of a virus like HIV. And when they looked at it comparatively, they came up with a hypothesis that if you look at the, where ARC exists in multiple species and its relationship to viruses, turns out the hypothesis that they proposed was sometime in the distant past, several 100 million years ago, a virus invaded the DNA of one of our distant ancestors. Instead of causing an infection, what it was domesticated. It was repurposed for a new use. See, these clumps in HIV, they are formed to help the genetic material move from cell to cell. Turns out these clumps in ARC are doing this. It's been repurposed to something doing similar in ARC. That is, ARC goes from cell to cell, and those clumps protect the genetic material as it goes from cell to cell. So a viral sequence was repurposed. And it turns out if you look at our genome, 8% of our entire genome were ancient viruses that invaded our genome, that have been knocked out. So our genome is a graveyard. Almost 10% of it is a graveyard for ancient virus and ancient viral infections. That's remarkable when you think about only 2% of our genome are our own genes, the sections that code for proteins. We have 4 times more viral genetic material inside of us than our own genes. And the story gets even more amazing. If you look at the placenta and some of the proteins that are active in placenta, guess what? They arose from viral invasions as well. And the more we look, the more we find. We have this very complex relationship with viruses. Now, this talk isn't entirely about viruses. This talk is about the surprises of evolution, that nothing ever begins when you think it does. And when you look at some of the great inventions of evolution, some of them, such as the gene ARC, certainly didn't begin where we think they did. They began as viruses. Now, this approach to evolution, I'm going to shift a little bit to talk about, return to Lillian Hellman's quote, nothing begins when you think it does, because it really explains some of these great transitions in evolution. What you see here is a slide of really kind of my own research, honestly. I study and find fossils about, that show us how fish evolved to walk on land, an event that happened about 375 or so million years ago. Now, when you look at this, transition from a fish on top to a land-living animal on the bottom. These are cartoons of fossils from the ancient fossil record. You think about the long list of features that have to arise to make this happen. It seems almost impossibly long, right? You have to have limbs, fins transform into limbs. Fish don't have necks. The first critters that walked on land like us have necks. Lungs, all kinds of inventions. They have to come about independently, separately at this transition. And if you make a list of all the things that have to change, that have to arise at this time, it would be impossibly long. But that's not how evolution works. Nothing ever begins when you think it does. And it relates to a story of one of the greatest characters in the history of science, Bashford Dean. He had two of the greatest jobs on the planet. I was jealous of Bashford Dean when I learned this. He was the curator of fossil fish at the American Museum of Natural History in New York. And so he was responsible for curating the fossil fish collection. He was particularly working on armorfish. At the same time, he was curator of the armor collection at the Metropolitan Museum of Art across Central Park. He was the only person ever to hold curatorships at both those museums and actually working on armor at both. He was working on armored fish in the American Museum of Natural History. He was working on actual armor and so founded the collection there at the Met. In the context of his work, he was very interested in fish and very interested in fish and how they breathe. And he discovered, and sort of rediscovered, because it was known in the comparative literature for a while, but made a very big deal of it, that a lot of fish have lungs. Look at this fish. This is a lungfish. This is Australian lungfish. There are actually today 3 different genera of lungfish living in southern continents. There's Australia, South America, and Africa. This is the Australian one. I think that gets the word for world's cutest fish. Let's face it, it's an adorable fish. But it's adorable in not only in its looks, but it's adorable because it has both lungs and gills. And what it does is it breathes with its gills when there's lots of oxygen in the water, when the oxygen content is high. When the oxygen content of the water falls low, what does it do? It uses its lungs. It goes to the surface and gulps air. And when you look at the structure of this fish's lungs, guess what? Identical to ours. They have 2 lobes. They have alveoli. They have surfactant. All the goodies that are inside our lungs are inside the lungs of these lungfish. In fact, all 3 genera of them. Turns out lungs aren't limited to these. Lungs are also seen in other kinds of fish. In fact, lungs are very common in fish. And when you look at the evolutionary distribution of lungs in the fish record, it turns out lungs are primitive. Yes, lungs evolved eons before creatures ever took their first steps on land, and they arose to help help fish live in water that had variable oxygen content. And that, as we look, the more we find that pretty much every invention that was necessary for animals to walk on land actually arose in fish living in aquatic ecosystems. This is Tiktalikrosia, a fish with arms and legs that my team and I discovered in the Canadian Arctic back in 2006. It's a fish with a neck. It's a fish with fins, but when you crack open the fin, what does it have? It has bones that correspond to upper arm, forearm, even parts of the wrist, even stubs of fingers. Really remarkable. And what this and other fossils show, and when we add in Bashford Dean's work, turns out that lungs, wrists, arms, necks, all the features that creatures were going to use to walk on land already arose in fish living in water. So nothing ever begins when you think that, though. They begin well earlier. And in fact, the great revolution in the history of life of fish evolving to walk on land wasn't as much about inventing new features. These features were already there in these fish. They were using them to live in water. It was repurposing them, changing their function. It was exploiting opportunities for allowing them to live on land when the need arises. And that's not this kind of thing. Nothing ever begins when you think it does. This is the rule. It's not an exception. If you think about birds, I can do this for every transition. Think about birds. Here's a mandarin duck. Wow, that's beautiful, huh? But it has feathers, like every bird. Birds have feathers. Birds have hollow bones. Birds have specialized lungs. Birds have wishbones and so forth. They have wings. Guess what? Each and every one of those features, including feathers, first arose in dinosaurs living on land. Feathers didn't arise to help animals fly. They arose in dinosaurs as they were living on land, presumably for thermoregulation, some sort of insulation, but also for maybe coloration patterns for courtship displays and other functions. But we know it wasn't flight. So the big shift, right, in the transition from land to air in this case, wasn't as much new inventions. It was using old inventions in new ways, repurposing. That's what evolution is about. Now, this repurposing extends all the way to the DNA. Think about this. This is where we all began, single cell, the fertilized egg. Now, that is really remarkable because that fertilized egg contains much of the information needed to form an adult body, a 4 trillion cell body. So going from the single cell, the fertilized egg on the left, to a fully formed adult body with trillions of cells all packed in the right place, we call this bodybuilding, right? Right, we call it bodybuilding. Anyway, there's an enormous amount of amazing biology built into the transition from the thing on the left to the creature on the right. And when you think about how a body is built from a single cell egg to a 4 trillion cell individual, you have a DNA recipe inside the egg that unfolds. And it unfolds in its interaction with the environment. It unfolds based on literally the computational information inside the DNA. And one of the things we've been able to do in science is being able to understand what the DNA recipe is, the genetic toolkit is that builds bodies to go from a single cell to a 4 trillion cell or more adult. And so I want to close with some insights on this because it's truly, truly incredible. And we now live in an age where we not only can sequence genomes, but we can manipulate the genomes using CrisprCast genome editing. We can edit the genome using a variety of different techniques. So one of the great surprises that came about, I'd say about 3 decades ago, is that there are a number of genes that are active in our bodies as we go from embryo to adult, as we go from egg to adult. And these genes are turned on and off as development proceeds. And you can see them here. You can see a human embryo on the bottom and a human on the top in yoga position. It's done by my artist who loves yoga. Anyway, but these genes define the basic architecture of our own bodies. Okay, where the limbs are placed, what the structure of our vertebrae are as you go down the vertebral column, where organs are in our body, where they are placed. Guess what? It turns out that other creatures have versions of these genes too. Mice, fish, flies. If you look at a fly body with its basic architecture, with wings in the right place, legs in the right place, and so forth, they all come down to certain genes being active and turned on in development. Versions of the same genes that build the bodies of flies are present in us, building our bodies. And these details get really, really beautiful. And I'm going to get into some details here. We're going to drill into some genetic experiments, and just follow me here because it's really worth. So this is Parhyale. Okay, Parhyale is a creature known as an amphipod. It's about, a centimeter long, like that. Okay, little tiny creature. They're called amphipods because they have lots of little different legs. Look at the legs in this critter. And you have the red arrow pointing to legs facing backwards. You have the blue arrow pointing to legs facing forwards. And if you look at them, you have a wide variety of legs. Turns out the genes are turned on and off to put those legs in the right place. I'm not going to go into the details, but scientists have been able to use gene editing technology to change the genetic address in the body to literally move legs around at will in the embryo. So if you treat the embryo early, you can actually make it so that it has all legs facing backwards instead of some legs facing forward and some legs facing backwards. You can have them, you can change it almost at will, rewrite it, using these genes that I'm telling you about. Well, we have versions of these genes too, and people have done experiments, not in us, but they've done it in mice. And guess what? It shows very similar things. Now, this is somewhat detailed, but let's look at this for a second. Here's a mouse. Okay, and you can see a mouse embryo in the middle, and let's zoom in on the mouse tail. Okay, so in the bottom here, you can see in the mouse tail, you have 2 types of vertebrae that we're showing you. You have lumbar vertebra on the top and sacral vertebra on the bottom. That's where the red arrow is. Well, guess what? You can do CRISPR-Cas genome editing on the embryo of a mouse, just like in that little amphipod Parhyale, doing versions of the same gene. And what do? When you manipulate them, you can transform the identity of vertebrae in the embryo as it goes to the adult. You can turn sacral vertebrae. You can make a mouse that has no sacral vertebrae on lumbar vertebrae. You can move the limbs around. You can change these things at will. So the basic toolkit that builds bodies like of creatures like us and mice is shared with worms, flies, and fish. It's a really beautiful story. And indeed, all the tools that are used to build our body originally arose in flies and other creatures. So nothing ever begins when you think it does. So you can ask the question, what's the relevance here? What the relevance is for all this is that we have deep connections to the rest of life on the planet. And if you look at the Nobel Prizes in medicine and physiology, excuse me, if you look at the Nobel Prizes in medicine and physiology that have been awarded over the past several decades, who have they gone to? They've gone to people working on flies. They've gone to people working on mice. In fact, 2 Nobel Prizes have gone to 4 people working on a worm the size of the size of a comma on a piece of paper. Truly remarkable. Yet studies of that little worm are telling us about how our genes are turned on and off in health and disease, and how our genes are programmed, our cells are programmed to die naturally, and what goes wrong in diseases like cancer. I like to think that as we discover cures to everything that ails us, from Alzheimer's to different cancers, that the breakthroughs that will extend and enrich our lives will in some way be based on a knowledge of flies, worms, and in some cases even fish. I can't imagine a more powerful or more beautiful statement on the importance of our evolution and our shared connection to the rest of the species on the planet than that. Thank you very much. I'm happy to take your questions.
Hi, Neil. So thanks, this is a great talk. So we've got a lot of questions coming in. Please keep asking them as we go. I'm going to try to keep one eye on the Q&A. So I have a quick science and entertainment exchange question just to kick it off. I'm curious, what are some tools that you would be excited for humanity to harness in our lifetimes from sort of evolutionary biology? I know that sort of gene editing and CRISPR are sort of possibilities, and a lot of filmmakers immediately go to the island of Dr. Moreau, but
yeah, definitely. Go to my lab, we do it there too. I mean, we have some pretty crazy fish that we generated in there.
So what are some exciting things that you think might happen in our lifetime with this new set of tools that we have?
The thing I'm most excited about is, inside our skulls, is a several pound little, trillion cells that can see the universe, that can go back 13.7 billion years and see the future, the human brain. And I think the more we have imaging technologies, CRISPR-Cas genome technologies, molecular technologies, behavioral, and understanding of behavior, the more we can unlock the secrets of the brain. And that to me is just, I was going to say mind blowing. I don't want to go there, but I think we're at the cusp of some amazing science to understand, the essentials of the brain, mapping individual cells, how they work together, how they work to form consciousness, memories, learning, and so forth. So yeah, I'm returning to Jason Shepard.
So you opened with the idea that 8% of us are these sort of this viral graveyard that sort of exists in our past. What do you think that can teach us about this current pandemic that we're experiencing today?
Yeah, that's pretty rough. So there's different kinds of viruses. So there's viruses that actually enter the DNA, okay, and commandeer the DNA that way. There are others that are RNA, a different molecule, that commandeer the machinery to control the genome. And so coronavirus is an RNA virus. It's a different virus. It's not one that actually enters the genome itself. That being said, let me just offer a few insights. It blows my mind that a little tiny piece of genetic material wrapped in a protein can change the world, right? Think about that for a second. I mean, if you ever want to be humbled as a human being, just think about how a little piece of RNA, and that knows not a lot of genes, wrapped in a protein with some lipids can change the entire planet. The couple things about when I just talk about coronavirus that the technologies that we're deploying to study evolution, the technologies that we're deploying to study the genome, we're very fortunate to be at a time to have those, because the genetic tools that we have nowadays means that science, which would normally take months to years, can happen in days to weeks. So if you look at the pace of science in the past several months, the technologies, computational technology, imaging technology, molecular technology, genetic technology, these are the things that are really changing, they're changing the pace in something which science can do. When you look at our own genome and our, the virus is in there, what you find is inside of us is a war going on. Our bodies, inside our bodies is a battlefield. It's a battlefield in our own genome and in our own cells between invaders and our own bodies. So invaders are always invading, viruses, bacteria, and so forth. And some of those invaders actually enter our genome. But our body responds to those. Now that war can have many different outcomes. One of those outcomes of that battle is actually fuel for evolution. And that's what we're seeing with the virus story. So it's, the battle can produce, new inventions. In fact, much of what we have in our bodies with that war of invaders and our own bodies has led to really kind of new pathways of evolution. I should say that war goes on inside our bodies, even with our own cells. I mean, cancer is a disease of our own cells. It's mutations in our own cells. And there's a war going on inside our bodies between selfish cells and cells that, support the whole body. So yeah, just think of our bodies as a battlefield at every level. And think about that as having multiple different consequences. It's complicated. In some cases that could be, can be bad, but in other cases it can be good in the sense of be fuel for evolution.
So over the course of your career, or maybe we could limit it to 10 years or some shorter period of time if that's easier to think about, what have been some of the great surprises for you in your research?
Yeah, there's been so many surprises. I think our human evolution story has just changed so much, and largely because of DNA work over the last decade and a half. If you think about, what the world looked like, say, 50 to 100,000 years ago, there are multiple different kinds of human beings walking the planet. Some of them interbreeding, some of them not. That is, to me, remarkable. Think about it. Think about having multiple different kinds of humans, species of humans, or subspecies of humans. And that's what existed, and that's what the genome technology is doing. So I think unlocking the mysteries of Neanderthal DNA, being able to sequence that, being able to see these human populations such as Denisovans and others, which they were able to isolate from DNA from individual bones. To me, the human evolution story, particularly the DNA-based one, has been really remarkable. But also I think the whole field of how genetic change happens in evolution has been just game-changing for us. Because you can imagine two different ways that evolution can happen genetically. One is a change in genes. That is, you change a gene, you change a protein. That's kind of how you think about it. But there's another way. Another way is changing the activity of genes, changing when and where a gene is active. That way has been one of the biggest gratifying surprises. And there's so much of evolution comes from changing the activity of genes, sort of the software of development, if you will. And so there's the hardware and software of genetics, and really the software changes have been very important, changing the kinds of changes that change the activity of genes. In my mind, that's been huge. But technologies have really changed the way I work in the last 15 years, remarkably. And it's not just genetic technology. Imaging technology. We now have CT scanners that can blast inside rocks, which for a paleontologist like me is awesome. I collect a block like from the Arctic, we can CT scan it, we can see inside it, and we can begin to remove the fossils digitally. That's been a game changer for us because now we can see inside the fossil record in ways we couldn't before. And imaging technology has gotten so powerful for all fields. So it's a really wonderful time to be a scientist in many ways, because there, every couple months there's new. It's also intimidating because it's kind of hard to keep up with all the change. But it's just access to questions that were always mysteries now we can unlock in many ways.
So one of our Director Retreat alums, Bronwyn Hughes, hi Bronwyn, says, what is the holy grail for you in your career from here? What's something that you hope to find before the end of your career?
Yeah, so Operation Next in Shubin land. So I've always had it in my head to find fossils that tell us about the great steps in evolution, right? And so the team discovered this thing. This is Tiktalikrosia. This is that early. It's a cast. It's not the original. Was one of the first fish to take steps on land. The one I showed you, it has arms and legs. What I'd really like to do is find the first vertebrate, first creature with a skeleton in the fossil record. The big transition is between a worm-like creature and a fish-like creature. That is huge, right? Because in that transition, you have so many new kinds of things that happen. Whole new biology comes about. For me, that means expeditions back to the Arctic, and rock's about 500 million years old. But yeah, I'd like to find the first creatures with skeletons. I'd like to find, skeletons like ours. And that's not just an esoteric question, because so much important biology came about at that transition, and we know so little about it that it's a natural attractant to me. So that would be, one of the things I'm really interested in. Then the other thing, I'm just come totally oblique at you here, is the problem of regeneration. And this is one thing my lab had shifted to right before we closed down for coronavirus. Now we're getting back into it. If you take a salamander and it cut off its limb, or it gets cut off naturally, it gets bit off or something, that limb will regrow entirely, okay? You'll get bones, muscles, nerves, vessels, fingers, everything back. We can heal wounds. That thing can heal like whole arms and legs and hearts and spinal cords. The problem with regeneration is really huge. So my lab's been getting into that. And so the holy grail for me on that one would be, how to think about how we can rebuild organs that have been lost. And I'll just give you one little insight that one of the people in my lab discovered, which is, turns out lots of creatures can regenerate, not just salamanders. Fish can do it and so forth. And if you look at the evolutionary distribution of regeneration, it's not like they gained it. Regeneration is primitive in creatures that live in water. We lost it. So why do we lose it? What do we need to get it back? So that to me is, that's another one. So origin of vertebrates, origin of regeneration. That'll keep me busy for a number of decades, I think.
So can you sort of first define the term to the audience of de-extinction, and then maybe wax poetic a bit about how you feel about it as an enterprise, and if there is indeed a creature that you would like to see de-extincted, what would that creature be?
Well, I'll go into that. The second one first, okay? I've been working on this one for 15 years, the fish with arms, Tiktalikrosia. I would love to see it in the flesh. What color was it? How did it behave? How did it move about? I've been spending a decade and a half making inferences from its bones and its fossils about how it moved about. It would really, really, I think I'd cry if I saw it, and walking around in silver. Really, I think. But so if I had to de-extinct anything, it would be this Devonian age creature from rocks 375 million years old. So when we talk about de-extinction, it means really, using kind of Jurassic Park, using the tools of the genome to bring back creatures like the dodo or the Tasmanian wolf or the mammoth and so forth. Now, when you look at the number of problems that have to be solved for that, you're really not going back. You can make you can sort of bring back a woolly mammoth in some form, but it really would be more like a hairy elephant. It may not be an original mammoth, because when you think about it, there are about 4 billion molecules that compose a piece of DNA. You have to get them all right, and it's more than that. It's the whole developmental context in the egg. So actually getting a creature back 100%, no, it'll be it'll be a copy, if you will. It'll be, rather than a mammoth, it'll be a hairy elephant. Which doesn't mean there's nothing we can learn from that. We can learn plenty from that. We can learn a lot about biology and so forth. There are ethical concerns, of course. But I'd like to see the dodo. The poor little dodo was hunted to extinction, and it's a beautiful little bird. It'd be nice to treat it well and bring it back if we can. But it'll probably be just like a big fat pigeon. It won't be entirely a dodo, because the complexity of these developmental systems is so large that you're not going to bring it back with high fidelity in toto. You'll have you'll have a version of it, but it won't be the version that existed 500 years ago.
So few people can speak about their work with as much passion as you clearly have, and so I'm wondering, what were some of the first moments that got you into science, that got you into paleontology? What was it that captured your imagination?
Well, I'm the last person you'd imagine to be leading expeditions to both poles. I work, I live in tents at, minus 30 degrees in Antarctica at 7,000 feet, leading a crew of 6 people, in nylon tents. I didn't camp till I was 20 years old. Okay. I never camped. I grew up in the suburbs of Philadelphia. I'm, the last guy you'd ever imagine would be camping, let alone leading expeditions to both poles. So what happened was I knew I liked science as a kid, but I liked archaeology. I loved astronomy, loved geology. I'm just a science kind of nerd. And when I went to college, I just saw in the course guide a course on human evolution. I'm like, oh, human evolution, that's cool. Maybe I'll take that. So I took this course first semester freshman year, and it was taught by a curator at the American Museum of Natural History. That's cool. But he'd bring in every week casts of fossils, and sometimes real fossils of primates and things. And I'm like, you got to be kidding me. You can go to places around the world and find things that tell you about millions of years ago. I was like, yeah, it really blew me away that you can actually do that, not just what it tells you, but that you can do it. And so I asked to volunteer in the museum. I volunteered, and then I had the world's most boring job. I was labeling stuff in drawers and stuff, which was fine. I enjoyed it, but it was really kind of more interactive with scientists. They'd be coming back from Argentina and China and the American West, and with stories of expeditions and new fossils to tell us about these great events in the history of life. I wanted to do that. So I got invited on an expedition in my junior year of college, and I was so bad, so terrible at it, that the head of the expedition, another curator at the American Museum, said, stick to museum work, son. You're just going to break more fossils than you find in the field. Forget it. But I really loved it. And I didn't give up. So I decided I wanted to do it. And so, I just stuck to it. So it's not where you begin, it's where you end up. But, and I'm not done yet. But it's that was it. It was I had more passion than talent, let's put it that way, initially.
So we have a number of questions about sort of the process of a gene turning on and off. And I think they can sort of be summed up a little bit by saying, like, when does a gene turn on and off, and how, and insofar as we're able to edit, what are sort of some moral safeguards that you think need to be put in place?
Yeah, there's a lot to unpack there. Okay, there's a ton to unpack. That's probably another hour-long talk. So let me get into it. So think about it this way. Here, let me tell you a little bit. So I'm sitting here with 4 trillion cells. You guys are all sitting there with 4 trillion cells. Each one of those cells, whether it's in your eye or your finger or muscle or whatever, has the same DNA, mostly the same DNA inside it. So what makes those cells different is not the DNA that's inside those cells, it's which genes are turned on and off in each of those cells. Right? That's what's doing it. So it's that computational machinery that's doing this. And so during development, as you go from egg to adult, genes are by the thousands are being turned on and off. It's like a concert of genes being turned on and off and interacting with one another. Now, there's a lot we know about this, and there's a lot we don't know. What we know is that DNA, that 6-foot-long strand of DNA, it's crumpled up into a ball. But that ball opens and closes and twists and turns. It's like doing a game of Twister inside the nucleus, right? And as it does that, as it opens and closes, it opens up that the genes in that open area become accessible, and they're accessibly turned on. So what happens is you'll have regions next to each gene, which are like switches, that when a molecule attaches to them, the gene is active, okay? And so sometimes what will happen is a piece of DNA will loop over and touch that switch next to a gene and cause it to be active or cause it to turn off. So there are switches throughout the genome. Think of them as molecular switches that other molecules attach to, and when they attach to them, they control the activity of the gene. And so really understanding this it's a complex network of these switches and factors in the genome, factors outside the genome, the 3-dimensional geometry of DNA as it opens and closes to do all this stuff. And all that's happening inside our cells right now. And it happens during development, remarkably. And we now have the technology to see every single gene turned on in a cell. And we can map it across an entire organ. That's the technology we have now. It's truly remarkable. We can tell each cell from each other by which genes are turned on and off in them. Every single one, sometimes thousands of them. And that really helps us to understand development and things like that. Now, genome editing technology is really powerful. CrisprCast is the one you probably read about. It was originally a bacterial sort of immune system, basically with a system that viruses would try to invade these bacteria, and then the CRISPR-Cas system would cut up the virus. But what is, it's been repurposed by scientists to edit genome. You can basically cut and paste genes, you can rewrite them and so forth. And there's a couple things that make it really powerful. The first is it's easy to do. I can train an undergraduate in my lab in about a week to do CRISPR-Cas genome editing, and we do it, and we do it in fish. It's fish embryos. The other is it works on a lot of different species. Not every species is equally well, but it works on a lot of them. I mean, it works on mammals, it works on mice, it works, works on invertebrates of all kinds of different. So it's incredibly easy to use. It's very powerful, and it's not expensive. We can do a run pretty cheaply on it. So that's why it's been so successful. Now, it's not 100%. When you edit a gene, remember there are 4 billion molecules, strands, bases in the DNA. When you edit one gene, you may try to think you're targeting a single gene, but there's a lot of other ones that may look alike. And so you may have what are called off-target effects. So, and we have a lot of those when we do genome editing in fish. We're targeting one gene, but there are other genes that may look like it elsewhere in the genome that we're affecting as well. That leads to a lot of things. You don't want to do that in humans, in the germline, right, in reproductive line, because you would cause all kinds of unpredictable mutations, off-target effects. So people are using CRISPR-Cas genome technology for a lot of things: agriculture, understanding evolution, development, disease. It's been used for making COVID-19 tests or SARS-CoV-2 tests for the virus. So it's incredibly powerful, but I don't think we're at the point where doing germline editing in humans, from adult to embryo that would go from generation to generation. I don't think we're in a place where we can do that, or should. However, you can do CRISPR-Cas genome editing on cells that don't, like for instance, skin cells that aren't, that are patch of skin cells that may have a disease state. Maybe you can do it there, or in adults and so forth. That's a whole different kind of CRISPR-Cas editing, but it's very powerful stuff. And we can edit the switches too. So we can actually edit the genome switches and control the activity of genes during development, rewrite it.
So jumping into sci-fi for a second, could you envision a couple questions around this? A world where we're basically, altering or enhancing folks for space travel, and could you envision a world where that would be useful in some of the ways that you might try to do that?
Yeah, I mean, you can envision lots of different kinds of changes. So for instance, with space travel, there's all kinds of problems that have to be solved, particularly long-term space. You can just make a long list of those. Now think about it, if you wanted to use CRISPR-Cas right now for that sort of thing, it would be very hard, again, because the off-target effects largely, but also because we don't really fully understand which genes always to change, because any given trait, any really interesting trait, it's not just one gene behind it, it's a whole battery, sometimes hundreds of genes. It's lots of little changes in different genes that together make the change. So you have to change whole batteries of genes, which would be really challenging. But that being said, if you think about the kinds of technologies we have to alter human performance, CRISPR-Cas genome technology theoretically is one, in the future, perhaps. But there are others, you think about devices, computational devices. Think about the cybernetics and robotics where you can, attach a robotic arm to somebody and they can have sensation related to that and motion controlled from their thought. You can have brain machine interfaces that are very powerful that can extend our reach in many ways. You have augmented and virtual reality systems that can be put to use. And there are therapeutics. It's not just the genome that is the basis of change, is what I'm saying. You can change the adult state through drugs, through machines, through brain machine interfaces and so forth.
So back to nature and evolution and the real world for a moment, not speculating on sci-fi. Can you give an example of something in evolution that occurred quickly and give a sense of what quickly means, and then something that occurred over a very long period of time? What very long period of time?
Yeah, so let me give you one that's a long period of time. The transition from fish to land living animal. We see the stages of that unfolding over about 15 million years, 10 or 15 million years, which is a long time. That's a lot of time. The places where evolution can really move fast, super fast, are when you introduce a new predator into an ecosystem, and that predator is like good at its job. It can cause change overnight. Think about the African lakes with the giant perch in there. So you can have introductions of species like that were formerly separated. When you put them together, the change in that can be within a generation or two, which is you'd never pick it up in the fossil record. It would appear instantaneous to us. So those kinds of things, you can change the world overnight. Invasive species, for instance, when you take two species that were separate, you put them together, and we see it in invasive species now with Asian species and the Great Lakes, for instance, in the West. You can get very rapid changes. I'll tell you what's really interesting is when people study evolution in the laboratory, when they take, say, flies or fish, and they do natural selection or artificial selection in the lab, what they find is that almost every system can change very rapidly in the lab, much more rapidly than we see in the wild. So the kinds of evolution we see that can happen in the lab, organisms are capable of much more rapid evolutionary change than we see in the wild. And the reason for that is in the wild, there's lots of different pressures that creatures have to respond to. You're not just selecting for one thing, like in the lab artificially. They got to live in an environment with predators and food and all that sort of stuff. And so that makes things go a little slower. So if anything, evolution can go a lot faster than we see, in the fossil record. But the kind of changes I work on take tens of millions of years, typically.
Do you think there are more zoonotic diseases in the world today, or do you think? Is there any way of knowing whether or not there's been an increase or is about the same?
Hard to know. If you look, so much of human history has been written by those diseases, plagues, and people coming in contact. The question is it because of, they didn't have as many therapeutics and they see more change then? But it's been a part of our history since the beginning. these kinds of diseases. Do we see it more? It's easy to imagine why you would, right? Because of air travel, because of human-animal contact, much more frequent, because of changing ecosystems, climate change, and so forth. You can imagine many reasons why it would be, but I don't know if the evidence that has taken more, but my guess would be more. We'd be crazy not to prepare for more of them, let's put it that way.
So I think this is going to be the last question in this segment. We, of course, do have the other Q&A that some of you signed up for in advance, and you should have been sent a link to that if you did sign up. So the last question is, can you talk a little bit about some fossil remnants, some lost functions or things that were tried and didn't quite catch on?
Oh, they're all kinds. Yeah, they're all kinds. So there's this creature called stripterus. Big name, big creature, 15 feet long. It was found in the, rocks pretty much the same age as Tiktalikrosia, this one. It also had a version of digits, fingers inside. It had fin, but it also had versions of fingers inside the fin. But these fingers were flat and they were branched. They weren't like single ones like this. They were forked and so forth. It had forked fingers, and it was just an experiment with its own stripterus with its own little forked fingers, but it didn't do it, So it turns out when you look at evolution, at some of these key moments, you'll see many different experiments with different similar kinds of inventions, and there's usually only one winner. Like in this case, was the Tiktaalik-like creature that digit-like arrows. But there were others that had versions of that as well. They just didn't make it for whatever reason. Probably not as good enough. Probably a fin not good enough to walk with. Let's put it that way. Then there's a long list of features like that.
Well, thank you so much for taking this time today, and we look forward to seeing you in the other Q&A session. This extremely readable book. Thank you very much, and we really appreciate it. And there's Anne. So time to wrap this section.
Exactly. We'll see you in the other room, Neil. And Rick, you're about to say something. I can see that.
I'm so sorry. We got 94 questions.
I know.
I'm really sorry if we didn't get.
But this is why we're offering this special sort of after event Q&A. We know because we get messages from a lot of you saying how frustrating it is that you've asked your questions, probably a really good question. And as Rick said at the top of this, we have an excruciatingly short period of time in which to take those questions. And as you can see, Neil is just such an amazing speaker with so much knowledge that I think we could all sit here and ask him questions for a really long time, and I think you'd continue to come up with really good answers. So I would encourage you, when we offer in the future the opportunity to have the separate Q&A, there will be a lot fewer of you in that other Zoom room. So the chances of getting your question answered go way up, and you'd be able to be on screen. You'd be able to pose your question directly to our speaker, and that's one of the reasons that we have decided to do this. And as Rick said, it's for a very small donation. It's not the $100 velvet rope. It's like the $5 velvet rope. So that makes it pretty easy.
Before we go, we should tell you next week's event, Science Speed Dating. Yet again, not actual dating. So please come in. You'll get a smorgasbord of science. Anne, is there anything else you want to say about that?
Well, we have, so far we have 4 confirmed speakers. Jasmine Drake, who is a forensic scientist. We have Emmanuel Taylor, who is an electrical engineer. Juan Gilbert, who was on our voting committee. And Kiki Jenkins from ASU, who is a very accomplished scientist, but wants to talk about the way in which she expresses her science through dance. So that should be really interesting. And we have another speaker that we'll be adding. We're just waiting for confirmation. And then and I also would say that we're showcasing diversity in next week's event. It's a lineup of all Black scientists, because diversity in science really matters. So we want to demonstrate the way in which these diverse voices contribute to our understanding of science. We also, for the following week, August 5th, we're back with the second in our series with our partners at the cultural programs of the National Academy of Sciences, COVID, creativity in the time of COVID. And so we'll send you more information about that, and we hope you can come there. The last last event of the season will be August 12th. We're going to be doing a screening of a documentary called Human Nature, and we'll be doing a talk back with the filmmaker and several of the scientists that were in the film. So we're going to wrap for the summer on August 12th, and we'll be back in the fall. But we hope that as you continue to receive your invitations, that you can make it.